primitive streak forms an elevation, the primitive node or Henson's node. Soon, a depression appears in the primitive node known as primitive pit. These cells from the primitive pit invaginate between the ectoderm and endoderm, migrates cranially, forming a solid cord of cells in the median plane known as the notochordal process. This extension is stopped by the precaudal plate, which is a small circular area of columnar endodermal cells where the cells of ectoderm and endoderm are fused together. This cellular cord, which extends between the primitive node and the precaudal plate, is known as the notochordal process. Soon, the notochordal process is canalized to form the notochordal canal by the extension of the primitive pit into the notochordal process. The cells in the floor of the notochordal canal becomes intercalated with the underlying endodermal cells of the roof of yolk sac. The fused layers gradually degenerates in craniocaudal direction, results in the formation of openings in the floor of notochordal canal. Thus, the notochordal canal communicates with the yolk sac. Through the primitive pit, the notochordal canal also communicates with the amniotic cavity. Thus, a temporary communication between yolk sac and amniotic cavity is established. This communication ensures the nutrition to the ectodermal surface of germ disc till the formation of blood vessels. The remaining part of notochordal process becomes flattened and forms a flat plate of cells known as the notochordal plate. This notochordal plate gradually folds along its long axis and separates from the roof of the yolk sac, which is now lined by a continuous layer of endoderm. The cells of the notochordal plate begins to proliferate in the cranial caudal direction. These proliferating cells enfolds and forms a solid flexible cord of cells known as the definitive notochord. The notochord functions as primary inductor. It stimulates the development of primitive vertebral column, and it induces the overlying ectoderm to thicken and differentiate into neural tube. The notochord degenerates with the beginning of the formation of vertebral body. The remnants of notochord remains a nucleus pulposus of intervertebral disc and apical ligament of dens. Formation of neural plate. During the development of notochord, the surface ectoderm over the developing notochord thickens to form an elongated neural plate. The neuroectoderm of the neural plate gives rise to the central nervous system. In the early stages of development, the neural plate is the same length as that of the notochord. Eventually, it extends cranially as far as the oropharyngeal membrane and extends beyond the notochord. Formation of neural groove and neural folds. The neural plate invaginates along the central axis to form a longitudinal median groove, which is known as the neural groove. Each of the lateral sides of the neural groove has raised lateral margins called the neural folds. These neural folds are prominent at the cranial end of the embryo. The tips of the neural folds are lined by specialized neuroectodermal cells known as the neural crest cells. Formation of neural tube. The neural folds move towards each other and fuse, converting the grooved neural plate into a neural tube. This fusion occurs in craniocaudal direction, and it soon separates from the surface ectoderm as the neural folds meet. Until the fusion is complete, the cranial end and the caudal ends of the developing neural tube are open, forming the anterior neuropore and posterior neuropore, which communicates with the amniotic cavity. The anterior neuropore closes approximately by 25 days and posterior neuropore closes by 28 days.